Well, I think we are all set. Welcome to the final panel before the cocktail reception. We are so glad there's more than 4 of you in the audience, so thank you for joining us. We are here to talk about what's going on in tissue engineering and talk about some of the latest innovations in repair and regeneration. Now, my name is Caralynn Nowinski Collens. I am the co-founder and CEO of Dimension Inc. We are a tissue therapeutics company combining cells and biomaterials to restore organ function, focused today on liver failure and other severe diseases. And I am super excited to be joined by my friends here on the panel. We all sit together on ARM's Tissue Engineering Advisory Committee, and it's been just a pleasure to get to know these gentlemen and the really exciting work that they're doing. And so we'll start with some introductions. And Geoff, would you kick us off?
Great, absolutely. Pleasure to be here. Thanks, ARM, for hosting this event as well and talking about tissue engineering. Geoff Ross, president of Miromatrix, where our mission is to bioengineer transplantable organs to eliminate the organ transplant waiting list. Our main focus in this therapeutic space is liver failure as well as renal failure. So our 2 top targets are liver and kidney. And earlier this year, we're excited to announce our IND to initiate our phase 1 clinical study for our MiroLiver ELAP, which is an external liver assist product where we actually take our bioengineered liver outside of the patient to treat acute liver failure. So really excited about the field, where we're going, and how we get there.
Geoff?
Good, thank you. Sorry, we have 2 Geoffs on the stage here, so I'm the Geoff. Really a pleasure to be here with everybody. Name's Geoff Hamilton. I founded Stemson Therapeutics in 2019. We're based in San Diego. And the mission of the company is to develop regenerative cell therapies and tissue therapies to treat alopecia and hair loss. So we're attempting to regenerate hair follicles to help folks that are suffering from that condition. And we're both a clinical and preclinical stage company. And as it relates to this topic today, we have this very exciting preclinical stage program that is attempting to create what we call bioengineered hair follicles. It's meant to replace follicles that have been entirely lost, either through damage or destruction of the skin on the scalp, or through just simply long-standing late-stage hair loss that has been going on for decades inside the patient. And so we deploy a number of different tissue-related technologies, engineering techniques, and other topics that we'll talk about today to create this bioengineered follicular unit that we are hoping to bring into the clinic in the next couple of years. So looking forward to telling everybody more about that, and thanks for being here.
Petter?
Hello, my name is Petter Björquist. I'm the CEO of VERIGRAFT. So I have a background of more than 25 years in regenerative medicine. The last 10 years with this company, obviously in tissue therapies. I'm very excited to be here in this panel with such great people. Thank you for coming. So my company is located in Sweden, so I'm a little bit of the odd sparrow here. My company is having a specialty, and that is to personalize tissues or grafts for transplantation. We are mostly active in the area of cardiovascular diseases. We are a clinical stage company, so we have our first product in clinical trials in Europe. So we are targeting a disease called chronic venous insufficiency, which is a disease caused by failing veins in our groin region. So that is something that we are curing in our trial, and we do see exciting data coming in as we speak. I'm very excited to speak to you about that today.
Right. Well, very glad to have you all here. And so we're here to talk about tissue engineering, which is something that is not new. Tissue engineering principles have been around for decades. This is the idea of bringing cells and biomaterials together to engineer tissues. And you know, we were all remarking and talking about this panel that we share a common belief that cells aren't enough. Now, we hope the best for our colleagues who are developing cell therapies, but we also recognize in our particular applications, cells need the right environment to be able to restore the functions that we're talking about, whether that's in the cardiovascular space, in the liver space, or for hair loss. And so today we want to walk through what are some of these new innovations in how we bring cells and biomaterials, whether natural biomaterials, as you'll hear in some of the cases of my colleagues here, or in the case of others in synthetic biomaterials. And so think of it as an old technology with a new look, and we are excited to highlight some of these new innovations. And so to get into a little bit of the science, I'd love you to all chat a little bit about what makes tissue engineering, what does the tissue engineering allow you to do that you couldn't do otherwise in order to get your products to patients? Maybe, Petter, do you want to start us off on that?
I'm happy to do that, thank you. So to give you a little bit about the background of the problem so that we are facing and that we are trying to solve with tissue engineering. So when we transplant, we are facing two major problems today. One is, of course, the availability of many organs or tissues. The other challenge is the immune system. So when we transplant, we have to put the recipient on lifelong immunosuppressive agents, and that is a real challenge for our patients. So our solution is to personalize, and that we do with tissue engineering. So we are exactly as Caralynn are saying, we are combining cells and something that cells needs to work in a good way. So on top of that, there is always a challenge for all of us in this field and in the entire field of regenerative medicine, and that is that we often are facing high cost of goods or significant cost of goods hampering this field. So we have a unique solution to all this that I described, and that is to source, instead of sourcing cells directly from the patients, we are sourcing blood. And using blood together with a scaffold, we can, in a very lean and quick and cost-efficient process, be able to personalize a graft for transplantation in as little time as 10 days. So tissue engineering is really something that we need in my field in order to offer the patient something that works and that can cure the disease. And we have found this unique solution with the blood. In blood, we find a lot of good things that we need in order to make this process efficient and working. So the solution for us is this process, and tissue engineering is our tool in order to make these two components, the scaffold, I will talk a little bit more about that maybe later, and the blood from the patients together give us a graft that can be transplanted and cure a disease.
And in your case, what we're talking about is the need for both of those components to be able to get the job done. We're talking about creating these venous grafts for the patients. We can extract the cells and other factors from the blood, but without having that scaffold to be able to repopulate, I'll say, and create a tissue-like matrix or a tissue-like product. You need both of those components.
Yes, exactly.
Yeah, right. Geoff, do you want to maybe build on that with
how
you guys are thinking about this?
Yeah, thanks very much. So, from our perspective, and maybe more broadly on tissue engineering, this is an applicable approach in areas where the endogenous functional tissues in the body are simply not there anymore. They're gone. They've either degenerated away, maybe they got destroyed through an injury. In our case and in our application with hair follicles, that's very much the case. For many cases of hair loss, you can get scars, burns, wounds. You can have chemotherapy destroy the hair follicles where certain patients simply don't have any functional tissue left. Or maybe you just have age-related hair loss that's been going on for 25 years and the tissue is completely gone. Point being, there is no druggable target in the body anymore from a traditional drug perspective. And so what you have to do is replace that tissue. So therefore, you have to kind of construct a tissue. It has, I think, in all of our experiences over decades, proven that typically you can't easily take cells and simply, if they're the right kinds of cells, if they're the right target cells, simply squirt them in the body and say, go figure it out, guys. I hope you build the structure that we want you to build. You have to, I'll call it, precede them, right? You have to give them some structure to enable the cells to start to self-organize. That could be at a small scale, like in our case with these engineered follicular units. We tend to find that simply starting, and we actually require more than one cell type. A hair follicle is a rather complex organ-like unit. Even though it's an appendage of the skin, it is a complex functioning unit. And you need several cell types to start talking with one another to self-organize. And we can start that process by preceding follicle organoids with, say, 20 to 50,000 cells. That's very small scale. Some other folks need a much higher scale, maybe hundreds of thousands to millions of cells, but you have to give them some sort of scaffold-like structure to begin with to enable a starting point for the tissue to then survive in vivo. And then typically, I think a lot of us, this is the case with us, we have a sort of a progenitor organ that we're putting into the body that then has to develop over weeks and months into a more functioning organ that we would see in normal adults. And so, it's a really exciting time to be in this space because we're seeing the convergence of a lot of different technologies to not only enable us to engineer functional cells, but then bring in biomaterials and other construction methods to enable these sort of organs to come together. So, it's really exciting to see it all come together.
So in your case, we're talking about organ-like, small, very small, organ-like
Very
structures.
small.
And
Yeah.
tens of thousands of cells. I saw, Geoff, you smile as that came up, as we're probably talking billions of cells as
We're
you're
talking
talking.
hundreds of millions to
Yes.
billions, so I'd love to
Yes.
be in the thousands business for a little while. I think to answer your question a little bit, Caralynn, on that too, is I think what tissue engineering, at least from our aspect, allows us to do is go after these large unmet needs. I think cell therapy and some of the things that they look at it, can you prevent the organ from being damaged? But once an organ's damaged, how do you actually develop a therapy that then can be a replacement? And we heard Scott Gottlieb, talk yesterday, and I certainly lived through this, of the promise of stem cell therapies when they first came out, right, late 90s. They were going to be the panacea. We just grabbed this high potential. All of us, many of us have been trained in developmental biology. We just let that thought fly right out of our head with this notion that we could inject it into a disease state and they would magically just cure and regrow the organ. Would have been beautiful if that would have happened, but I think in hindsight, it's not a surprise that it's much more complicated than that. And I think what tissue engineering offers us is that ability now to take the appropriate scaffold, grow the tissue that we need to, and measure that level of functionality then to be able to place that back inside the body to actually create therapies that are replacement therapies for this. And we've got vessels, right? We got blood, not blood, vessels, we got hair. And then certainly what we're going after is how do you engineer something very complex like a liver or a kidney using a biomaterial. In our case, we use a natural biomaterial, a porcine matrix that we decellularize, and then we infuse that with human cells to be able to bring that functionality back together to be able to create that therapeutic that should have the overall functionality then to become a replacement therapy.
Yeah. In all of our cases, I like to think about it as we need to get enough cells to go where we want them to go, to organize in the way, as you said, Geoff, you can't just like squirt them in and they're going to organize. You need to give them a way to organize. And then it needs to function quickly and over the long term to be able to provide that sustained durability, that ultimately that tissue engraftment that's necessary for this kind of function.
Yeah.
So I want to bring you all back to the beginning of your journeys for a moment. And let's talk about which came first, the cells or the biomaterials? At what point did you recognize that this needed to be a more holistic approach? And maybe let's start in the middle, Geoff, with you on this one.
Yeah, I love this question. I mean, in our case, it was clearly started with the cells. And I think anytime you're trying to replace a tissue, that is, I'll call it the primary mechanism of action. The cells have to be able to function and organize. And only through testing and, advisory and things like that did we learn that you have to provide some sort of structure and that biomaterials, for example, could be a good way to do this. In our use case, we also have difficulty in things like handling. We need unit level control to make hair look normal. And if we're trying to create, 10,000 individual units, it's very important to be able to sort of handle and transplant in a very specific way. So first it was sort of, came the cells, a method for being able to produce the highly functional target cells that have the folliculogenic potential that we need, that is actually not easily obtained from adult cells, we learned. And so getting to the target was sort of the key. And only afterwards, as we got into what I'll call a little bit out of the biology question and more into the engineering question, did we learn that we probably need to add a scaffold. And I think our preclinical in vivo results have demonstrated that scaffolding, that biomaterials approach, we also use some natural biomaterials as well that are conducive to the skin environment, really started to enable us to produce the individual follicular results we wanted. We were quite good at producing these awful looking hairy balls of tissue underneath the skin, all of which were ingrown hairs. And I don't think any patient would want that in any case. And so that's where the materials came in second.
Yeah. Right. Now, Petter, you come from materials background.
Yes. So in our case, the scaffold has been around for decades. We are using a technology called decellularization. I'm sure you are familiar with that. That is basically to take a scaffold from a human body or from an animal and you decellularize, meaning you take off all the cells. So that has been known for decades. So that was not really the challenge. Our challenge was the cell source. And we were facing back many years that people were isolating cells from the patient. A typical way was to take bone marrow out of the patient, to identify the cells you need in bone marrow, you isolate those cells out, you put them in culture, you expand them. This is a process taking months, up to 4 to 6 months even, before you have cells enough to do the second process, the personalization process. So we were thinking about a way of doing that, and we found that the blood itself is a perfect material, also sort of a biomaterial. And you should remember that blood is not only cells, that is also a lot of non-cellular components, platelets, a lot of proteins that we can use in our personalization process. So for us, the scaffold came first, and then we invented this unique technology that my company is mastering that is now covered by more than, I think we have 82 granted patents around that process now. And that is why we can produce now to much lower costs and in a very lean way that regulators love.
Yeah.
Well, Geoff, I mean, this is kind of a perfect segue because you're using similar old technologies and making them new again.
Yeah. I mean, for us, it's certainly the biomaterial came first. And I think it's because as you look at how do you solve a big challenge, like how do you bioengineer an organ? And the first thing that you really come across from that standpoint is what's your supply? And during development, our vascular system forms almost immediately because diffusion only takes you so far, maybe a millimeter, if you're really, really pushing the boundaries. So if you want to create a solid tissue or a really advanced tissue, you need an intact vasculature, or you need some way to be able to get the nutrients to the cells. You got to remove the waste, and you got to have that dynamic nature. So for us, it was really being able to solve this, and our approach is similar to Petter's approach, which is decellularization. In our case, we use perfusion decellularization. We actually take whole organs from a pig, cannulate them, perfuse a mild detergent through them to rapidly remove all the cellular material. It leaves you with that perfect scaffolding. Now, early on, we did have to solve, how do we know that porcine is going to be an appropriate source? How do we know the body's not going to reject that? So we actually took whole porcine livers, perfusion decellularized those, and we created a surgical mesh and a wound care product that we got 510K. We commercialized that. Thousands of patients have been implanted with the matrix itself. So it was a really good way to de-risk it as you go after something complex like this. and then start adding the cells back onto it now after the functionality, knowing that your base biomaterial, at least you got the safety profile for it, but you also have the vasculature, and now you've got a structure that can be able to support this high density of cells and this complication of cells at that level to move forward with the product side.
Yeah. So as you know, we too started on the biomaterial side of things at Dimension. In fact, our first product is also an acellular product that's in patients for bone regeneration. And it's a device because it doesn't have cells. It is just in fact, it's synthetic biomaterials that are 3D printed into a structure that then fills a bony defect. And so we took that through the FDA, through CDRH, not through CBER, because we were a device. And I want to kind of start talking about how all of us are thinking about, these are products. These are products that are going to be in patients. And CBER is going to be the one that's regulating all of these products, including where we're also going at Dimension and defining biomaterials and hepatocytes for liver failure. Yet the modality looks a lot like a device. So tell me about your challenges, or maybe how you've thought, if you've thought about this as an opportunity, you can do that. But Geoff, maybe you want to
Yeah, I mean,
start?
as you talk through that, I'm like, hey, are we just a glutton for punishment? Because, I mean, first we try to tackle something difficult. The cell side's not hard enough. Let's throw a biomaterial in there and characterize the, interactions. But then when you move to the regulatory side, we're all going to be combination devices. So it's not one agency. Now we're dealing with two agencies. So I think we just really like to tackle complicated products. I think from our standpoint, certainly, we commercialized that. CDRH was the same agency that we worked through for the 510K. When we then moved over on our IND for our external liver product, our bioengineered liver, that is CBER, but it is a combination device at that standpoint. So because the mode of action is the cells, CBER is really the lead on it, but we do have consults in CDRH from that aspect. So you do have to be cognitive of, on the regulatory side, as you move through that, how that changes, but also how all that knowledge needs to be kept together to be able to really enable that product to move forward.
Yeah. Well, maybe if you can talk for a moment on kind of the evolution of your company and kind of what that meant for you ultimately finding a partner with United Therapeutics and how you're thinking about that, because a lot of the companies that we might naturally look to partner with don't necessarily know, well, frankly, they don't know what to do with us.
Yeah, I think we've talked about this a lot. I mean, certainly we've had side conversations as part of our committee and stuff like that. And sometimes we're a tweener, right? And what I mean by that is our challenge is you heard us earlier, we're talking about how do you create therapies that are going to cure large unmet needs, right? These are not going to be cheap therapies at that standpoint, right? They're going to be in line with cell and gene therapies as you look at that. Broad stroke, some of these are going to be a big range of that, right? So it has the potential from the low end all the way up to the high end, depending on what value you're adding. But all of us on this stage have a transplant component to it. So while you may have markets that are similar to pharma markets and not traditional device markets, your mode of delivery and where this will be delivered in your patient side is much more surgical, like a traditional device. So we often get in these conversations, right, of the challenge of this, of kind of being a tweener where people don't really know where to put you. I would say that hasn't stopped any of us of how you move forward. I think continuing to focus on what's the problem that we're trying to solve, what is that data? Ultimately, data drives all of this. So while people can't sometimes see how is the end market or how is that going to be served, the closer we get with data and demonstrating the efficacy of our therapies, the rest will continue to follow.
Yeah.
I would just add real quick that while being a tweener is always not the most fun place to be, I think it provides an opportunity for those folks in this area to sort of usher in this. I mean, I genuinely would refer to it as a bit of a new modality. And yes, data will trump all, but once we sort of cross that valley of death, it's, I think, true for any sort of new therapy in the space trying to make it and trying to demonstrate efficacy. Once you get that data, I think the standard players, not just the regulators, but even like big pharma who can drive a lot of the funding and pathway forward for companies like ours, will stop looking at us with a quizzical look and start really, I think, jumping on board, because they do, a lot of folks understand the huge unmet need in these spaces. And certainly patients are not quiet about their unmet need. And I think we're starting to see real momentum across this space of tissue engineering. There are more and more companies that are, I think, coming onto the scene that I think can fairly represent for tissue engineering, and we sort of need more to come forward. But I think we're, the fact that we're even here on stage, where we wouldn't have been 2 or 3 years ago, thanks so much to ARM for the support there, I think is an indicator that folks are really starting to understand that, not that just that this new modality is coming onto the scene, but that we're really starting to build some real successes and some interesting sort of early days of patient successes and stories, which is really the key to this whole thing.
Yeah, I mean, I completely agree. I mean, if I go back 5 years ago, the number of people I talked to who said, look, this is too complicated, it will not work, you'll never get this through the FDA, there is no path, right? That's what's exciting about this field right now, is we're able to continue to move forward, and now you're seeing INDs cleared for complex products based on true data that demonstrates, that you're willing to be able to go to that next level. So now there is a path, a regulatory path, for these very revolutionary therapies, everybody up on this panel included, which I think is really exciting for the field in terms of where we're at.
Yeah, well, and Peter, you're in the clinic now, and so actually treating patients and working with the European agencies in this case. But
So maybe I'm jumping between the questions now, but to connect to the first thing here, I'm from Europe, and in Europe this is quite clear. So my company has never considered transplanting only the material side of it. So we have always had cells around. And then the European authorities are saying if you are manipulating cells, you are a drug. And with the drug here is we have exactly the same classification as a cell therapy. So that is quite clear cut. Going to the next thing here, I think it may be a confusion that tissue therapies and tissue engineering always are very complicated, because I think my company is an example of when the cellular component can be actually simpler than cell therapy alone. And together, I think we have lower cost of goods than many cell therapy companies are having. So I think there are so many innovative solutions and so many ideas how we can generate the material component or the scaffold component and the cell component. Imagine how we can use printing today. So we are seeing our next generation of products being based on a scaffold that is printed, and that will lower cost of goods. That will make everything much simpler, and together with a cell component that is really simple, I think we are a good example of a tissue therapy that is really something less complex than people believe.
I'm so glad that so many of you brought up the idea around the cost of goods, because we too believe that we are able to provide some efficiencies, some economies by being able to perhaps lower the number of cells that are needed, because we're able to keep them more localized within a scaffold approach, or be able to, promote additional expansion or differentiation through the use of a scaffold. So there are these economies that are coming as well. And I think, Geoff, as you said, we're talking about a new modality here, or one of you said that, I'm sorry, one of the Geoffs said this. One of the Geoffs said this. We're talking about a new modality here, and, you all heard us talking about this idea of tissue therapy. And I think, even the FDA, and thinking about advanced therapies broadly, is grouping the work that we're doing as part of that mandate. And so as we think about, where tissue therapies are going, and not just thinking about this as just cell and gene. In a couple of years, there's going to be C, G, T, and T, right? We're going to and T and T. So I'm going to come up with a new acronym. ARM folks, taking note. But as we think about where this is going, we've talked about that. Maybe it's not as complicated. There is a clear path. What are some of the things that make you excited about the future here? What is giving you encouragement about where we're going?
I'll start off for just a moment. Maybe I'll point to two things. One is some emerging clinical data about how tissue engineering approaches can solve what I think are probably otherwise unsolvable problems through other modalities. And so I think very excited to see what happens in both of your clinical trials as it relates to that. What I'm also seeing, I'll point actually to what I think is a really positive new emerging event on the regulatory front is a few countries are really waking up and starting to prioritize regenerative medicine. And I'll just give a shout out to our partners in Japan and on the regulatory side. I think this was sparked by the discovery of the induced pluripotent stem cell capability out of Japan in 2007, and the Nobel Prize on that in 2012 is they recognized that these cells now these iPS cells have the ability to generate any target cell type of your choosing and therefore open the possibility to regenerate whole tissues, because we now could get access to a source of cells that you can't get. And a lot of adult cells, for example, are not programmed or good to actually generate new tissues. They've already gone through that during our fetal stage, for example, and they just don't do that anymore when they're in adult stage. And so Japan has offered a relatively, I'll call it relatively smooth and rapid path to, as long as you can demonstrate safety, to lower the bar quite a bit in terms of efficacy required to at least get on to market, to get to commercialization. And so I think this is starting to maybe goad some other regulatory agencies to think hard about how to enable some of these types of products to come to market a little bit faster and to get the clinical data that we need quite a bit faster. And I think that's really opening up everybody's eyes on how these different tissue therapies can make a big difference in patients' lives, especially in really, really high unmet medical needs that can't be addressed otherwise.
Peter, do you want to?
Yeah, I can move on. I realized that I jumped away from your question about the fact that we're in the clinic now. So for me, when you talk about excitement, it's easy because, I have the world's best job for the simple reason that I'm a biologist. I love biology. I do biology all the day in one way. But at the end, we also see that we can do something really good for patients in need. So we went clinic 2 years ago. And the very first patient that we treated, that was a patient that couldn't even leave the apartment because of the serious problems caused by the disease. And this patient told his doctor that I have a dream that once again go to a football game. And football, that is soccer here, right? So go to a soccer game. And that sounds like a little thing. But this patient was really burning for soccer and had a dream to go once again to a soccer game. And we treated the patient, and 2 months later, the patient went to a soccer game. And those type of stories is making my life fantastic. And I think that's why we're all here, because we are dreaming about doing things like that. And in my case, we cannot do that in any other way than using tissue engineering.
I mean, that's why we're all here, to ultimately get to patients. So, Geoff, I imagine you're excited about also getting in the clinic, but what is encouraging you about this space?
Yeah, I think certainly on the patient side, I mean, it's just encouraging to start thinking about developing therapies that can help patients. I mean, if you look at some of our patient populations, there's 500,000 patients on dialysis in the US alone. 90,000 are on the transplant list, but only 24,000 this year are going to receive an organ. So, I mean, it's exciting to see how we're making progress against some of those targets and offering people, a different therapy, a different option that's outside of what's available today. I think certainly on the regulatory side, I'm really excited about the regulatory progress that we've seen with the super office, with some of the other things, and just we're seeing these therapies move forward. We're seeing the openness to collaborate, to talk. And understand some of these complex therapies. With that said, I mean, this is all built on, the knowledge of cell therapy and everything else. So we just see the whole field continuing to be lifted up to the point that you can start advancing some of these other therapies. So I think that's exciting. I mean, I think the last thing is, and you hit this a little bit indirectly, but we certainly do that, and that is, you know, we know with most therapies is the quicker you can get them back in the body, the better, because the body is the ultimate bioreactor. We have not been able to emulate that bioreactor outside of the body, but I can say with some of the current technologies with AI, you're starting to see a stronger understanding of what are the culture environments, how can we really control those? Because ultimately, while it is we talk about this, you know, it's complex, but our job as therapy developers is how do you take something really complex, make it repeatable, manufacture it, and essentially remove all that complex nature of it. And I'm excited about the tools that are starting to evolve, and we're starting to see that actually enables us to do that, which I think allows this field to move forward too.
Great. Well, I can keep asking you guys questions, but I do want to make sure we open it up to the audience and see if there's any out there. So any questions from the group out here? I think there are some microphones available if needed. All right, I'm going to keep going, but you guys get ready. If you feel like you have one, raise your hand, and I'll be scanning the audience here. So ultimately, this is about getting it to patients. It's about getting our products to market, to patients, and that requires resources. And so maybe we'll talk to everyone's favorite topic, the funding environment, and how we think about getting the investment that's necessary. And unfortunately, we know that investment generally in cell and gene has continued to be challenging in this market. We add this, again, perception of additional complexity, but I think all of us would agree perception in that case. Maybe you can just talk a little bit about the kinds of things you wish maybe investors understood about this market, or maybe just some of your experiences as you've been thinking about the funds needed to get to the next stage. Peter, do you want to start us off here?
I can start. So you are right. We are in a tough period to finance companies like the one we are running. That is a general financial thing, but also maybe a little specific for this field. But I also see a positive trend. I see that us doing tissue engineering are able to explain that tissue engineering and tissue therapies are not as complicated as people are in general thinking. I mean, it's logic to think that cell therapy is quite a complicated thing, and if you have cells and something more, it must be even more complicated.
But it's biology, right?
It's biology, and I think we have to go back. If we can explain that we are solving a huge unmet medical need and can help patients with a severe disease with a tissue therapy, I think that is the first start. And then we have to explain that this can be done at reasonable manufacturing times and costs, etc. And I think we are on our way now to move that field and explain that tissue therapies is really something for the future.
Yeah.
I'll add, and by the way, I thought I saw a hand rise from somebody,
okay.
But let
Great.
me just say
I'll
one
be
thing.
right
and
there,
then
Tom.
We'll get to a question since we only have three minutes left. But I'll add sort of one of the interesting experiences I've had with investors and getting them comfortable. I actually rarely have a challenge describing the unmet need in hair loss. Half the people I pitch to say, please put me in your clinical trials. But demonstrating to them some sort of, I'll call it predicate, whereby some at very, very small scale that couldn't be scaled up into a product, some type of tissue was able to solve this particular problem in a patient or two. And if we can bring a clear technical approach that can then create similar or same tissue as what was used in that procedure, that can then be scaled and controlled and brought forward into the market at reasonable costs, that's a really helpful approach. And in our case, hair transplantation is the most common, what I would call organ transplant procedure performed in the world. About 200,000 people every single year in the United States get a hair transplant procedure where a doctor or surgeon just excises like a biopsy punch of individual hairs out of a donor area, typically the back of the head, and then brings them over to the target area. And where it's removed from, the hair never grows back, so you're not creating a new supply. But as you can sort of create that predicate and walk the investor through how your technology and procedures can kind of mimic that and mimic those results. For tissue therapies, I've found a number of other similar companies in the space have sort of explained how that's really helped them. I know that has had an effect in companies in the type 1 diabetes space as well as in the Parkinson's space as well too.
Continuing to get those modalities going out. So I'm going to pause here for a moment, and I think, Tom, do you have a question?
On that same theme, and you may have answered this question already, but you know, somebody mentioned stem cells in the 90s was a big thing, and homing and to the site of injury, and then tissue engineering was going to be the big promise before it all crashed down in 2002 with organogenesis and advanced tissue science and everything coming down. And it wasn't until CAR-Ts came around and tissue engineering became CAR-T's awkward cousin that everyone focused on CAR-T, and there's a huge amount of investment to fund all of this work. Maybe I'll put you on the spot and say, what is the CAR-T of tissues? Because I want to know, what do I focus on at Army? What do we scale? And what's that CAR-T of tissues that's going to get us to that point with tissues and not just immunotherapies? Recognizing that those have bled over into autoimmune and NK cells and all kinds of other stuff.
I mean, I think it's a great question, and you know, I was sitting here thinking about that a little bit too, and there's parallels to all the past therapies. I mean, I think if you study antibodies, right, monoclonal antibodies, it's the same case. Like a lot of hype, tons of investment, it dropped out, people thought it was dead, and then you see that reemergent, right? So you go through this valley of death where good ideas and good science wins. Eventually you see the products and then you see it come to fruition, right? We saw that with gene therapy. I mean, you saw the hype with gene therapy and where we were with that, right? It got out too early, set the field back by decades. Now you see the research and you see the technology working. I think we're on the same trajectory with tissue engineering. There was a lot of early hype, a lot of investment. We all saw the ear on the back of a rat, you know, and how you move these things forward. And now the tools and therapies are getting caught up. I think to answer your question, what's that for CAR-T? I think it's just products to get to market and to start seeing some of these. So you do start to see more products in advanced clinical studies. I mean, you know, Humacyte, well, that's an acellular graft. I mean, it's still the first one that you'll see going through a BLA and starting to, once we can develop these products, have the ability to get to patients, treat patients, and define that market, I think that's the CAR-T. And certainly any of our clinical trials that start to provide positive therapy, whether it's on whole organ, whether it's on hair, bone, vessels, I think any of that stuff's going to just give the field momentum to show that all of this therapy is possible, and it's going to lead to products.
I don't know that we can predict the killer app that will emerge first, right? I mean, you're seeing some exciting things happen in many different organ areas, heart, liver, I think, and cardiovascular. We'll just have to follow the data to see which one looks like it's going to be most clinically successful, but then I would also argue could be most scalable. And CAR-T's had its own scaling issues, but it's been scaled enough that it's, you know, now considered a big success. So we'll have to follow it.
Well, I think we better end on that note, but I think this group will argue the time is now. It's only going to get better from here in terms of the opportunities for these tissue therapies. So if you have any more questions for us, I hope you'll come find us in the cocktail reception and we can talk more about the advances in tissue engineering. So thanks to my panelists, thanks to the audience, and we'll see you next time.